Hi, my name is Russell Rockne. I am Director of Mathematical Oncology and faculty at City of Hope. I have here with me Andre Rodin, who is the Susumu Ono Chair of Theoretical Biology, and Peter Lee, who is a physician scientist and chair of the Department of Immuno-Oncology at City of Hope. Andre, can you give us a 30-second pitch and briefly describe the innovation? Who will it benefit, and what more precisely is the value being created?
So the 30-second pitch is this. Every research and medical lab that does flow cytometry or high parameter cytometry in general should use our algorithms and software because they could recover a great deal of information that has been lost or ignored by the conventional pipelines. Like, for example, FlowJar, which is installed in pretty much every single lab right now. So that was a 30-second pitch. Now, a little bit more about the value being created and added. First and foremost, this is multivariate analysis that can pick up signals. And by signals, we mean biomarkers that flow join basic gating plus clustering and visualization miss. And we're talking about not just single biomarkers, but their interactions and concerted action. And for that, we have the systems biology, the network analysis, and machine learning classification regression type analysis. So the emphasis is really on the predictive biomarkers discovery and not just the basic cell gating and clustering. And current alternatives on the market, both commercial and open source, they just don't do that.
I see. So you mentioned that the new algorithm and software could recover a great deal of information that is being thrown away or ignored by conventional flow cytometry like FlowJo type pipelines. Could you give us an example?
Well, our original interest in the field, it stems from an immuno-oncology study in which we looked at the gastrointestinal cancer patients that undergo checkpoint blockade immunotherapy. And we wanted to look at the immune networks, which we captured through flow cytometry data, FACS data of immune system snapshots in peripheral blood, and compare these networks, compare them before and after treatment and between responders and non-responders. We found the existing algorithms and software limiting, so we developed our own, centered mostly around Bayesian networks, which is a computational systems biology method. And we found a number of predictive markers, and biomarkers for response to the therapy in particular, very strong, statistically very strong signals that the conventional workflow like FlowJar simply missed. Well, obviously now we are thinking about generalizing our approach to other studies and other cancers.
Interesting. So Peter, could you tell us more about the immunotherapy study Andre just mentioned? How does it fit in the broad context of current immuno-oncology?
Yeah, so this is a clinical trial conducted by my colleague, Joseph Chow, who's a GI oncologist here at City of Hope. And it consists of 14 patients with metastatic gastroesophageal cancer treated with a combination of checkpoint blockade anti-PD-1 antibodies and also radiation therapy. And of the 14 patients, 3 had durable clinical responses. And so we had an opportunity to try to understand biologically what's different between the responders and the non-responders, looking at primarily peripheral blood, but also some tissue. And because this is a small study with only a few responses, it would have been very hard to see any significant differences based on traditional methods. So we're very lucky to be able to work with Andre and you to use these powerful new approaches to get high dimensional analysis and deeper insight into the data. And very pleased that we could see a very clear signal where we would not have seen that before.
Right, I see. So Andre, get for this interview.
Well, I would say that any lab really, especially in the immunology and immuno-oncology space, but not limited to that. So any lab that does high parameter flow cytometry of FACS. Well, obviously, algorithmically, this can be easily extended to any type of high parameter cytometry in general. The algorithms are the same. The data pre-processing routines will be different. So I would say a very broad market, really. And since we shared our first results here at the City of Hope a couple of weeks ago, we have already started 3 new collaborations with the investigators who are interested in applying this type of analysis to their studies to find something more sophisticated, I would say, in their data.
So that begs the question, Andre, are there existing competing products on the market? What are the main competitive products or services?
Well, I would say that the 2 most established software platforms are FlowJo and Spade, but they only do manual gating and clustering, what we call drawing pretty pictures, and are not really focused on delivering the actual predictive biomarkers and their combinations and interactions. Well, another example is Astrolabe, which is a recent product. It goes one step further than FlowJo in that it deals with raw data, and it does basic statistics on it, but it's still univariate. There are no multivariate network analysis, and most importantly, you cannot automatically infer and rank predictive biomarkers and their interactions. And in fact, one of the primary reasons we are having this conversation right now was that we had an Astrolabe presentation on our campus recently. And we looked closely at it, and our reaction was, oh, we can do so much better.
Absolutely. Peter, could you give us your point of view then on the potential market?
Yeah, so I've been involved in flow cytometry for 25 plus years. And so we started with 2, 3 color flow, and then that went up to 4 and 6 and 8 and 10. And now quite a few labs are doing routinely 20 something. We routinely do 30 color flow. And there are instruments, and we have one here as well, that can do even 40 plus colors. And so these are very high dimensional data that is, there are too many things going on with the interactions for the human mind to be able to keep track. And so the existing analysis tools really are more just straightforward. The user defines what to look at, what not to look at, and the software just basically helps to display the data, but there's not much of any intelligence behind those platforms. And so this is really the first time where there are algorithms to look at the interactions between data points and to help us come up with connections that we would not have been able to see on our own. So I think if we could really make that even more successful and more user-friendly, I think there could be huge potential here.
So then, Andre, are the algorithms and softwares patentable? How would one go about building a strong intellectual property position then?
Right. Well, the basic algorithms, some of them, for example, for building networks, they're open source. But more dedicated algorithms such as network comparisons and cytometry data-specific data processing routines are patentable. Well, we just published a paper with a proof of concept and a real-world example, but the core details and proprietary algorithms were not spelled out. So we are working on establishing an IP position on this. Perhaps even more importantly, we're also working on a set of new algorithms for handling simultaneously 2 levels of data, cell subtypes and markers per se. And once ready, these algorithms, they would be proprietary, certainly, and definitely patentable.
And so then what steps do we need to take or should be taken in order to bring the innovation to market?
Right. Well, first and foremost, I would say developing a strong user interface or a server cloud implementation. And we do need to do more algorithm development work to maneuver seamlessly between the 2 levels of data that I mentioned, cell subtypes and the actual markers. This is fairly straightforward. I mean, in practical terms, this means 3, 4 scientific programmers working for 1, maybe 2 years. We are actually funded right now by the NIH for the algorithm development work, the mathematical work, but developing commercial grade interface and implementation is a really whole different kettle of fish. What we have right now is a bunch of C++ and Python code. It works fine, it's robust, but it's not what you would call like a usable off-the-shelf solution. What we would like to have, and what we should have really, is an interface similar to, well, I don't know, similar to Microsoft Excel, really, but fine-tuned and targeted to the cytometry data from spreadsheet type data manipulation to biomarkers, network comparison, and visualization, like ready for publication.
So then, Andre, what type of commercialization opportunity is this? Is it licensing to an existing company, entrepreneurial startup, a partnership and sponsored research?
In general, all three. Again, repeating myself, this is just straightforward software development work that can be done under either of these mechanisms. But I would say that at this stage in development, either a partnership sponsored research or an entrepreneurial startup would probably be the best fit.